Okay, hi everybody. This is the last lab video podcast of the term over the eye and the ear. And remember, as always, do not use these video podcasts as a substitute for coming to lab and going to biosis. It's simply meant to help improve your performance on the practical, but not as a substitute for, again, actually interacting with the models and the dissections so you can become as familiar with the structures as you possibly can. So the first thing we're looking at is the eye model, and what we want to know is what is this layer of the eye called? This is the sclera. This is part of the fibrous tunic of the eye. It is largely avascular and protective in its function. What are these? These are the superior and lateral rectus muscles. The superior muscle, of course, is on the top of the eyeball, and when it contracts, the eyeball looks upward. The lateral rectus muscles will turn the left eye left and the right eye right. And then this is the optic nerve. Okay, this enters the back of the eyeball, forming a spot on the retina known as the optic disc, or your blind spot. What's unique about that is it has no photoreceptors. The reason we don't pick up a blind spot in our normal field of vision is because the right and the left eye don't share the exact same location for their blind spot, so that when you look with our stereoscopic vision, it appears as if we have a complete field of view. In addition to that, the fact that the eyes are in front of our head and give us slightly different information when we gaze forward is one of the things that provides us with the depth perception we so desperately need if we're talking about a hunter-gatherer, which is what man was in his early development. To demonstrate this to yourself, you can take your index finger and hold it about a foot in front of your eye, okay? And what I want you to do is close your left eye and look at your finger, and then close your right eye and look at your finger, and notice how the background moves behind your finger and how your finger seems to move as you gaze at it with one or the other eye shut. The fact that this information is slightly different when you look at your finger with both eyes open, this is what gives you the dimension of depth in your visual field. Okay, this is the same model with the outer fibrous coat bisected away. Okay, what you're looking at here is the iris, which controls the diameter of the pupil, this little round area here, and it responds to light intensity. The greater the light intensity, the narrower the iris will become. The less the light intensity, the wider the iris will become. The more light is needed in dim situations in order to be able to see. Now, what controls those reflexes are smooth muscle that are located around the opening of the pupil that can either contract or relax and change the diameter of the pupil accordingly. When the pupil constricts, that's a result of parasympathetic innervation of these muscles, while when the pupil relaxes, that's a result of sympathetic innervation of these muscles. The clear skin in front of the iris and the pupil is called the cornea. It is unique in that when light passes through it, instead of scattering, it will bend. This is something called refraction. And then, of course, the opening is the pupil. And then finally, this dark layer here that lies just underneath the fibrous tunic of the sclera and the cornea, that's called the choroid coat. It's unique because it contains a lot of blood vessels and as a result nourishes the retina as well as the choroid coat. So what we need is choroid coat in order for the retina to remain viable. Now we've removed the choroid coat, we're looking actually inside the eyeball. This clear body here, this is the lens. Again, notice that it is transparent and that instead of scattering light, it will bend it as it passes through on its way ultimately to the retina. Down here, you can see the cornea again, and of course, this is the optic nerve coming into the back of the retina, and you can see the optic disc right there. Okay, that's where there are no photoreceptors. That is the point of entry of the second cranial nerve, the optic nerve. And this, of course, is the vitreous humor, which has a couple of roles. Number one, it maintains the shape of the eyeball, but number two, it forms a pressure seal that keeps the retina pressed against the back of the eyeball so that it does not detach. If the retina were to detach, you would lose that portion of your visual field, and you wouldn't be able to send that information back to the occipital lobe of the brain, which is where the primary visual cortex lies. Now we're looking inside the eyeball. We've removed the vitreous, we've removed the top part of the fibrous and the vascular tunic, and we are gazing here now at the inside of the eyeball looking out. So this, of course, is the cornea, followed by the lens, and then suspensory ligaments, which hold the lens in place, attached to special muscles called ciliary bodies, which can change the shape of the lens depending on whether we're focusing for near or for far. Essentially, when an object is close to your eye and you want to have it be in sharp focus, the lens has to become fatter in order to refract the light to the point where it'll actually hit the back of the retina and be in sharp focus. If you want to demonstrate that to yourself, hold your index finger about a foot in front of your eye and then slowly move it forward and try to keep that finger in focus. And as you do, you'll feel those muscles pulling on the lens, attempting to retain focus for the object you're looking at. Okay, so when the object's distant, it's a different story. The lens tends to be thinner and flatter, and as a result, it's a lot easier to see sharp edges on your image when it's far away. That's why in individuals that are suffering from a condition known as presbyopia, where the lens has lost the ability to change its shape depending on where you're directing your gaze, it's easier to see things far away in terms of distinct outlines than it is to see things close up. And so this is usually when we need reading glasses. And then, of course, the innermost tunic of the eye is the retina. That's where the photoreceptors are located. What they do is they transform stimulus in the form of light into electrical information that's relayed back through the from the photoreceptors to the bipolar and ganglion cells, and ultimately from there to the optic nerve and to the occipital lobe of the brain, which is where we process a lot of our primary visual information. Okay, so there's three tunics in the eye. Don't forget, the neural tunic is the innermost, the vascular tunic is the next most superficial, and the fibrous tunic, which is made up of the cornea and the sclera, is the most superficial. And again, you can see the optic nerve and the optic disc right here. Okay. Now, what are we looking at in this drawing? The round area, that's the pupil. That's what allows the light to pass through the lens and ultimately hit the sweet spot of the retina, which is called the macula lutea. If we look at the eye and all of its layers, we can see that light actually passes through four transparent layers on the way to the retina. The first being the cornea, the next being the aqueous humor, which is a liquid that lies between the lens and the cornea, and then the vitreous humor, which lies behind the lens and goes all the way back to the retina. And as the light passes through these layers, it will bend, and as it bends, it'll come into sharp focus, and in a normal eye, the perfect focus will be directly on the macula lutea, which is the site of the greatest concentration of photoreceptors, specifically the cones, which are responsible for color vision. Okay? This, of course, is the iris, which controls the diameter of the pupil. Remember that it is constricted in bright light and it is dilated in dim light. Next drawing here, you can see the optic disc. You can also see the retina. The structure, what structure fills the space behind the lens, and that's, of course, the vitreous body or vitreous humor. Now, that is essentially a clear jelly-like substance, which again has two purposes. It seals the retina against the back of the eye, and it maintains the shape of the eyeball, and it's also transparent so that light can pass through it. Now, a couple of things about the retina while we're looking at this view. There's only two places where the retina is physically attached to the eyeball. One place is this little sort of sawtooth border here. This is called the ora serratia, and this is the anterior attachment of the retina to the eyeball. And then, of course, it's attached at the back of the eyeball via the optic disc, which is again the point of entry of the optic nerve. The rest of the retina is only held in place by the pressure that comes from the vitreous humor. Okay, this is why if you were to ever lose some vitreous humor from your eyeball, you could have retinal detachment very easily. Now, next thing we want to look at is the sheep eye dissection, which we all did in lab. Again, the white fibrous outer layer is the sclera. The clear layer is the cornea. Now you might say, well, wait a minute. In this picture, the cornea doesn't look clear. And the reason for that is that the cornea has been sitting in preservative, which tends to dehydrate that portion of the fibrous tunic and turn it opaque so that when light hits it, it scatters instead of passing straight through while simply bending. Okay, so this is diffraction as opposed to refraction that you're seeing in this preserved cornea. Next up is the lens, and then down here you can see the suspensory ligaments, which are attached to the ciliary bodies, which again control the shape of the lens. Okay, remember that the lens tends to be fat when objects are near in your view, when you're trying to focus for near, and the lens tends to be thinner in shape when the objects are further away. The clear jelly-like substance behind the lens is the vitreous humor. This, of course, is the pupil. And then finally, the iris, which is what controls the diameter of the pupil. Okay, again, letting in more light when there are dim conditions and letting in less light when there are high intensity light conditions. This is just another view of the sheep eye. You can see here the cornea, the sclera. This, of course, is the lens, okay? And you can see also the vitreous humor. Now, the aqueous humor in your dissection probably leaked out of the eye while it was sitting in the preservative, and that's why you didn't see very much of it when you actually did your dissection. Moving on, here you can see a kind of an interesting layer here. The sort of yellowish stuff that you can see, the little yellow patches, that's retina, okay? Under the retina, that shiny part is called the tapetum lucidium, and that's a special iridescent layer that helps to increase the light intensity inside the eyeball so that certain animals can see at night, okay? So make sure to distinguish between the retina, which is a delicate yellowish layer that basically when you open the eye up kind of becomes detached. You can see it kind of down here a little bit, as opposed to the tapetum lucidium, which is the iridescent layer that lies underneath it. If you're confused about which layer they're asking for on the practical, again, raise your hand and ask a question. And this point, of course, is the macula lutea. This is the point of the greatest concentration of photoreceptors in the eye, okay? And this is where we get our greatest visual acuity. This is where the image comes into focus when we are properly focused on an image, okay? We want to put it in the sweet spot of the eye so that we get the greatest amount of visual information. This is the pigmented epithelium of the retina. The reason it's dark is to trap light inside the eye. You can think of the eye as a tiny movie theater, and we don't want stray light coming in from the outside or escaping the eyeball, and as a result, the black absorbs a lot of light, and as a result, the only light that matters is the light that's projected onto the back of the retina. No stray light bouncing around inside the eyeball to cause problems. This is again looking at the sheep eye from the back to the front, and you can see here the ciliary body, and you can also see the cornea, okay, and I believe you've got a lens there as well, okay, so we're looking back to front there. Okay, these are some histology slides looking at some of the sensory structures we're dealing with. This is a retina slide here, and what you're looking at literally is the back of the retina. This is the vascular tunic. This is the portion that contains blood vessels that help nourish the neural tunic. This is the fibrous tunic, the outermost layer, which again in the eyeball is composed of the sclera, which is the white of the eye, and the cornea, which is the transparent portion. And this, of course, is the optic disc. This is where the optic nerve, shown here, enters the back of the eyeball, and as a result, you can see there's no photoreceptors here. Okay, notice the lack of the ganglion, the bipolar, and the photoreceptor cells in this region. But again, all the visual information is poured from the retina into this nerve, and it heads directly back to the occipital lobe of the brain. So be sure to be able to identify that if you see it on the practical in a slide. This is simply a look at the retina. Here you can see the retina in total. Okay, this of course is the sclera, and we're going to break the retina down next. Okay, remember that light comes in through the cornea, through the aqueous, through the lens, and through the vitreous, and then it actually passes through the retina and hits the choroid. And it's on this first pass through the retina that the photoreceptors pick up the light energy and transform it into electrical impulses. And so the ganglion cells are the most superficial cells in the retina. They're the closest to the vitreous. The bipolar cells are the intermediaries between the photoreceptors and the ganglion cells. So the light is approaching from the direction indicated by arrow A. So what happens is the light passes through. Once it gets to the photoreceptors, which are here, they will change their membrane properties based on a chemical that's embedded in a photopigment called rhodopsin. Okay, and the active ingredient in there is called retinol. And what will happen is that the retinol, when the light hits it, will change its shape. That'll cause the rhodopsin to change its shape, and that will cause a change in the membrane potential in the photoreceptors, which will turn into a graded potential, which will ultimately become an action potential and be carried from there to the bipolar cells, from there to the ganglion cells, and from the ganglion cells to the optic nerve. Okay, so the front of the eye would be up here, the back of the eye, of course, would be back here. Okay, next thing you want to become familiar with are some visual tests, and what we're looking at here is a chart for visual acuity. What aspect of vision does this chart test? This tests for errors of refraction, specifically myopia. Okay, in myopia, what happens is that the eyeball is too long for the optical elements that come with it. Okay, and as a result, the image comes into focus before it gets to the retina, and the result is that when the image gets to the retina, it's blurred. And so we have to correct this by using concave lenses in the glasses that increase the focal length so that the image hits the sweet spot of the retina and comes into sharp focus on the macula lutea. 2040 indicates the visual acuity of the person taking the test. The denominator refers to the acuity of the normal eye, while the numerator refers to the acuity of the subject. A subject with a ratio of less than 1 has low acuity, while a subject with a ratio of greater than 1 has a higher acuity. Okay, so you can see here the different sizes of the letters are going to be proportional to the ability of the eye to focus on what's actually located on the chart. Okay, somebody with 20/20 vision has a ratio of 1. That's considered to be quote-unquote normal. That means that they can see at 20 feet what a normal eye can see at 20 feet. And as a result, they don't need any corrective lenses. Okay, so be aware of that when you see that on the practical visual acuity test. Okay. Next visual test. This is a test for astigmatism. question is, how might this chart appear to persons with an uncorrected astigmatism? And the answer is that certain lines will appear more clearly than others. An astigmatism is basically an irregular shaped cornea, which causes parts of the image to come into focus too soon and parts of the image to come into focus too late to hit the sweet spot of the retina. And we generally have to correct this either with laser corrective surgery or with bifocal lenses. What are the different types of defects that might cause astigmatism? Well, corneal astigmatism occurs when the cornea has an irregular shape, and lenticular astigmatism occurs when the lens has an irregular shape. Either one of these can cause the image to be blurred for reasons either of being in focus too soon or too late when it hits the retina. What term describes the changing of the shape of the lens when the eye is focused for close vision? And the answer is accommodation. Next question, does the lens become more convex or less convex when the eye is focused for close vision? And the answer is more convex. Next question, does the pupil increase or decrease in diameter when the eye is focused for close vision? And the answer is it decreases in diameter. And that's again to avoid stray light causing the image to become less distinct. The reflex demonstrated here is the pupillary reflex. Notice that the pupil is constricting in presence of bright light. Is this reflex a result of smooth or skeletal muscle contraction? And the answer is it's a result of smooth muscle contraction. Next question. What feature of vision is being demonstrated in lab using this card? This is looking for the blind spot. Okay, so the way that this works is that you hold the card about at arm's length from your eye, okay, and then what you'll do is you'll close either your right or your left eye, and with the eye that's open you'll focus on either the dot or the cross. It doesn't really matter. And then you'll move the card towards you, and at some point the other image, either the dot or the cross, depending on which one you're not focused on, will disappear. And that'll indicate exactly where your blind spot is in the eye that is open. Okay? You can practice that at home with a 3x5 card. Okay, now we move on to the ear. And we'll look first at the ear model. The ear, again, is a special sense organ, and its job is to convert stimuli in the form of sound into electrical impulses that are translated to the temporal lobe of the brain, resulting in an action potential that's generated by a very special organ called the cochlea, which you can see here, kind of cut away. And that is sent through the 8th cranial nerve to the temporal lobe of the brain, and that's where the auditory cortex is located, and that's what we interpret as sound. Okay, so this is the external acoustic meatus, and this is the pinna. Okay, the job of the pinna is to funnel sound into the acoustic meatus and guide it to the tympanic membrane shown here. Okay, a couple of other features. This is the tensor tympani muscle. It's responsible for causing the eardrum to become taut in the event that there is a loud noise. It's basically a fail-safe to avoid damage to the eardrum in the event that that happens. The stapes is the most medial bone of the middle ear, and it rests right on top of the oval window of the cochlea. This is the incus, and this is the eustachian tube, which connects the middle ear to the throat via the nasopharynx. And this is present in order to regulate pressure in the middle ear with the pressure in the external acoustic meatus, so that the eardrum isn't either sucked back against the ossicles or protruding outward from the ossicles as a result of pressure problems. This is the tympanic membrane, also known as the eardrum, and this is the malleus, which we sometimes call the hammer, okay, which is directly connected to the eardrum on the medial aspect. Okay, now the eustachian tube is also a route for bacterial infection of the middle ear, causing a condition known as otitis media or earache. The way this happens generally is if you have a cold or an infection in the throat and you blow your nose, you can force bacteria up through this tube and into the middle ear, where it will begin to grow and produce gas, and that'll push out on the eardrum, causing it to distend. This is terribly painful. The remedy for this currently is to treat the infection with antibiotics. The problem, of course, with this approach is that often by the time you have an advanced ear infection, the eardrum is close to rupturing, and often the buildup in pressure will cause the drum to burst. The fluid and the exudate will then flow out the external acoustic meatus, and the pain will be over. The eardrum will heal, but when it does, it'll have scar tissue, and the result of that is reduced auditory acuity. Now, in the old days, the way they would treat the earache is to lance the drum with a needle in order to relieve the pressure, and also treat with antibiotics. That's not common practice anymore, but in the event that your child or yourself get an ear infection, otitis media, if you take your anatomical knowledge, knowing that the middle ear and the throat are connected, and you want to relieve the pressure of an ear infection, if you close your nose and your mouth and you inhale as hard as you can, you'll create a vacuum, and you can pull some of the bacteria and gas down the eustachian tube and into the throat, where you can spit it out. So that's a temporary mode of relief. Depending on how occluded the tube is, it'll either be effective or not. But in any case, just something to be aware of. The reason children have more ear infections than adults is because their eustachian tubes are shorter and straighter, and it's an easier trip for the bacteria to get up into the middle ear. As we age, the eustachian tube becomes more convoluted. It's, as a result, more difficult for bacteria to get up into the middle ear, and so earaches are less common. Treatment for this in childhood is to insert myritogamy tubes into the tympanic membrane, which regulate the pressure on either side of the eardrum, and in the event that bacteria get in there, the oxygen that's provided by the myritogamy tube tends to retard the anaerobic bacteria that like to produce the pain of otitis media, and as a result, the frequency of ear infections and earaches goes down quite a bit. Okay, here you can see the cochlea. This is the organ of hearing. This is the vestibulocochlear nerve. Remember that that's the 8th cranial nerve. And these are the semicircular canals. They're responsible for our sense of balance. Okay, when we're moving, the semicircular canals tell us in what direction we're moving: up, down, left, right, back and forth. Okay. Down here is the vestibule. In the vestibule, there are structures that respond to our sense of static equilibrium, so that when we're standing still, we know top from bottom and left from right. They respond to gravity via sensory hairs that are embedded in a gel-like matrix that contains otoliths that are attracted by gravity, and the result is that we are able to maintain our equilibrium. This is the organ that allows us to stand up straight and to walk and perform activities in a purposeful manner. If we didn't have a functioning vestibule or semicircular canal, the slightest movement would produce disorientation, nausea. Okay, this is the organs of the middle ear removed from the temporal bone. You can see here the vestibule. These are the semicircular canals, and this, of course, is the stapes that rests right on top of the oval window. Here is the incus, which is attached to the medial aspect of the eardrum, followed by the malleus, which is the middle bone of the middle ear, and then finally the tympanic membrane, which is also known as your eardrum. So we're looking from medial to lateral in this view. Okay, it's as if we're standing in the middle ear and looking out towards the pinna. Another view, you can see here the vestibule, the cochlea, okay, the organ of hearing, and the sensory organ found within this structure is called the organ of Corti, and that is what actually transforms sound stimuli into electrical impulses that we interpret via the temporal lobe as noise or speech. What membrane does this bone rest against? Again, that is the oval window. Essentially, what happens when sound is transmitted to the eardrum is that the eardrum will vibrate, causing the malleus to hit the incus, which causes the stapes to vibrate, and that causes the fluid inside the cochlea to move. And as that fluid moves, it causes sensory hairs inside the organ to go up and down, and as they bounce up and down, they open ion gates, and this produces the electrical impulse that we interpret as sound, which is relayed via the cochlear branch of the vestibulocochlear nerve back to the temporal lobe of the brain. This is a cross-section of the cochlea, and you can see here the spiral ganglia, the tectoral membrane as well. Okay, these are the sensory hairs which, when the fluid moves through this organ, the endolymph here and the perilymph here, as they move through as a result of the vibration of the stapes, these hairs start to bounce up and down like so. Okay, and the result is that we open ion gates, we send information back to the spiral ganglia, and then we relay that eventually to the eighth cranial nerve and back to the temporal lobe of the brain. Okay. Here you can see the tectoral membrane. You can see the inner hair cells and the outer hair cells and the basilar membrane. The collective term for the sensory structure seen here is called the scala tympani. Okay. Now, I'm going to put a video on here that shows you how it is that the eardrum and the eye actually work. So stay tuned.
To form images of our environment, light emitted by or reflected from objects must be focused on the retina. Some of the light reflected from the surface of a yield sign is directed toward the eye. Upon reaching the eye, the light is first refracted by the cornea to send it on a path toward the retina. To reach the retina, the light rays must pass through the lens, where a small amount of refraction adjusts the angle of the light to form a focused image on the retina. Central to the sense of vision is the transduction of light energy into electrical energy by cells in the retina called rods and cones. Cylindrical-shaped cells called rods are responsible for the perception of light and dark. Conically shaped cells called cones are sensitive to red, green, or blue light.
The location of the eyes enables us to see length, width, depth, and distance of an object. The eye has three layers: the sclera, choroid, and retina. The sclera is the outer white layer that maintains the shape of the eye. Muscles attached to the sclera control eye movements. Choroid is the middle layer that contains the blood vessels. The cornea is a clear circular area in the sclera where light enters the eye. The pupil is the circular opening in the front of the choroid. The iris is the colored smooth muscle surrounding the pupil, which adjusts the size of the opening according to the brightness of light. The lens is located behind the pupil and between the anterior and posterior chambers. The lens is a transparent, flexible biconvex structure that bends or refracts light rays, so they focus on the nerve cells of the retina. The chambers are filled with a watery fluid that gives shape to the eye and helps refract the light rays. The anterior fluid is called the aqueous humor, and the posterior fluid is called the vitreous humor. The retina is the inner layer and contains the nerve cells, the rods and cones, and the bipolar cells. Rods are sensitive to light but do not sense color. Cones sense color. The highest concentration of cones is in the fovea centralis. The rods and cones synapse with bipolar cells, which then synapse with the ganglia cells whose axons form the optic nerve. Light rays enter the eye through the cornea, pupil, and lens. where the light rays are bent or refracted to focus them on the rods and cones, which transmit the stimulus to the optic nerve and then to the occipital lobe of the brain for interpretation.
In this video, I want to discuss some of the structures of the eyeball. I'm not going to talk about the muscles that move the eyeball, the extrinsic muscles of the eye. We've done a separate video on the muscles of the eyeball already. So on this view, I just want to take care of some of the layers of the eyeball. The eyeball is actually divided into 3 layers, also known as tunics. And from this view, we can see the outer white portion of the eye. This is called the fibrous tunic. And the fibrous tunic is actually divided into 2 different regions. The clear part of the eye, known as the cornea, and then the white part of the eye, known as the sclera. So the cornea and the sclera, both part of the fibrous tunic. Now here we've cut away a portion of the fibrous tunic so we can look deep to that. The next layer in is called the vascular tunic. And you can see here on this model depicted are blood vessels spread out on the vascular tunic. So its name is very appropriate. From this view of the model, we can't see more of this vascular tunic, but when we look inside the model, we will see more of that layer. The last thing I want to point out on this lateral view of this eyeball is the optic nerve. The optic nerve, cranial nerve number 2, is sensory for vision from the eyeball. It is slightly to the medial and inferior aspect of the eyeball. I have removed half of this eyeball so that we can see the other structures. Again, the 3 tunics of the eye. Here we can see the white is the fibrous tunic. Then we see a brown layer, which is our vascular tunic. And then we see a pinkish layer. That is the nervous tunic. Some books call it the retinal tunic, and it makes up the retina. If we come toward the front of the eyeball, again, we see the sclera and the cornea, both part of the fibrous tunic. The vascular tunic posteriorly is known as the choroid, and then as we come forward, it widens out into an area known as the ciliary body, and the ciliary body will have ciliary muscles which will control the shape of the eyelids. We'll talk about that briefly. And then it also extends forward so that the vascular tunic makes up the colored part of our eye known as the iris. So the iris is part of the vascular tunic of the eye. And then in the middle, we have the structure known as the pupil. The pupil is actually the anatomical lack of a structure. It is an opening in the middle of the iris. Directly posterior to the pupil, we see the lens of the eye. From this angle, that actually defines some different chambers of the eye. We have the anterior chamber of the eye, which is located between the cornea and the iris. That is known as the anterior chamber of the eye. It is filled with a fluid known as aqueous humor. Between the pupil and the lens, or between the iris and the lens, very hard to see on this model, is a much smaller chamber known as the posterior chamber. That chamber is also filled with the aqueous humor. It is in continuity with the anterior chamber through the opening of the pupil. Behind the lens of the eye, we have what is known as the vitreous chamber. The vitreous chamber is not filled with aqueous humor. It is not filled with vitreous humor. It is filled with a jelly-like substance known as the vitreous body. Vitreous body. Now notice on the posterior portion of this eyeball, right here we have a white spot. That corresponds to where the optic nerve entered the eye. There are no photoreceptors of the retina at that spot right there, so it is often referred to as the blind spot. Its anatomical name is the optic disc, also known as the blind spot, because any light focused on that portion of the retina will not stimulate any sense of vision. Now if I rotate this model just slightly, we can see as the retina comes forward and meets the portion of the vascular tunic coming back from the ciliary body, we have a serrated edge. That serrated edge is known as the ora serrata. The ora serrata. I want to take just a little bit of time to focus in right here on the ciliary body. In that ciliary body, I said there were ciliary muscles, and those ciliary muscles attach to small ligaments known as the suspensory ligaments of the lens. Those fibers will attach into the lens so that by putting tension or releasing tension in those ciliary muscles, we can control the shape of the eye lens. I will leave it to your individual lecture professors as to what they want you to know about that particular structure. From this angle of the eyeball, we can see some other things that weren't shown on the other side of the model. A quick recap though, the white is our fibrous tunic, the brown, our vascular tunic, here as the choroid, and then here as the ciliary body, and here as the iris. We can see the anterior chamber, the posterior chamber we have to imagine because we do not have the lens, and then the vitreous chamber. But I want to focus on the back of this vitreous chamber. Here we see somewhat of a brownish spot, more of a peach color. That is called the macula lutea. The macula lutea. And in the very middle of that macula lutea, we see a yellow spot known as the fovea centralis. The macula lutea is very good color vision. There are more cones in that area than there are rods. And in the very middle of that macula lutea, when we get to the fovea centralis, we have our very best focused vision, and it is mostly color vision. That area, that yellow spot, known as the fovea centralis. In this video, I want to talk about the extrinsic muscles of the eyeball. Extrinsic muscles of the eye are muscles that are not part of the eyeball itself, but yet go from various regions of the skull and the orbit into the eyeball to direct the movements of the eyeball. The first muscle I want to talk about on this model is really not an extrinsic muscle of the eye, but it is located on this model. It is a muscle which functions to move the eyelid, coming from the annular ligament, traveling in a superior and forward direction out to the eyelids. And on this model, I'll turn the model a little, you can see the gray portion represents the tarsal plates of our superior and inferior palpebrae, or our eyelids. We see the muscle known as the levator palpebrae superioris muscle. Its name tells you it lifts the superior eyelid. The levator palpebrae superioris muscle. It is not technically an extrinsic muscle of the eye because it does not move the eyeball, but is certainly associated with the eye. Now in this model, if I remove the tarsal plates of the eyelid, I can see that I still have my levator palpebrae superioris muscle, and immediately underneath that muscle I have another muscle which travels from the annular ligament out onto the anterior superior surface of the eyeball. That muscle is known as the superior rectus muscle. The superior rectus muscle functions to elevate our gaze, cause our gaze to look up toward the sky. If I remove the levator palpebrae superioris muscle on this model, it takes the muscle belly of the superior rectus muscle along with it. Now that I have removed the superior rectus, we can see into the medial side of the orbit of the eye, coming from the annular ligament and a little bit of the sphenoid and ethmoid bone, we see a muscle which travels in an anterior direction, and then if I tilt the model a little, you can see that it comes forward and it passes through a connective tissue loop on the frontal bone. It does not connect there, but goes through the loop and then travels in a posterior lateral direction across the eyeball. This muscle is known as the superior oblique muscle. The superior oblique. The name oblique means to travel at a slant, and we can see that as it comes across the top of the eyeball, it is running at a slant. The superior oblique muscle functions to rotate the gaze down and laterally, or down and out. On the lateral aspect, we can see running from the annular ligament straight forward to the lateral aspect of the eyeball, a muscle known as the lateral rectus. The lateral rectus. The lateral rectus functions to pull the lateral aspect of the eyeball posteriorly, which would cause our gaze to look laterally. We call that abduction of our gaze, to look laterally. Now if I remove the lateral rectus from this model, we can see in the posterior aspect of this model, or I should say the medial aspect of this model, the medial rectus muscle. Now I have to tilt it just a little bit so you get a good view of it because it's hidden behind the optic nerve. The medial rectus. The medial rectus muscle is just the opposite of lateral rectus. It will pull the medial side of the eyeball back, so that will cause our gaze to look inward, or adduction of our gaze. On the lower portion of the orbit of the eyeball, and I'll have to tilt this whole model, coming through the lower portion of the orbit of the eye, running straight forward from the annular ligament into the bottom of the eye, in an anterior-inferior portion, is the inferior rectus muscle. The inferior rectus muscle rotates our gaze such that we would look downward. So there are four rectus muscles: the medial rectus, the inferior rectus, the superior rectus, and the lateral rectus. And as their name implies, they direct the gaze in their appropriate direction. Medial rectus deviates the gaze medially. Superior rectus, the gaze will look superiorly or up. Lateral rectus, you look laterally. We call that abduction of the gaze. Inferior rectus, we look down. We already talked about the superior oblique. If there is a superior oblique, you know there must be an inferior oblique. On this model, if we look for the inferior oblique, it comes from a medial aspect right where the lacrimal bone and the maxilla meet, and its fibers travel underneath the eyeball to insert posterior laterally on the inferior portion of the eyeball. That is the inferior oblique. The inferior oblique functions to rotate the gaze up and laterally, or up and out. So the two oblique muscles rotate the gaze laterally. The superior oblique rotates down and lateral. The inferior oblique rotates up and lateral. So those are the 7 muscles that we consider to be extrinsic eye muscles on this model. Technically only 6 were extrinsic eye muscles. The 7th was the levator palpebrae superioris.
Yeah, my on-off switch came on. Okay, you're...
In beginning a dissection with any dissection, it's always important to look at the external features first before jumping in there with your instruments. You want to make sure that you try to see anything you can recognize here. I recognize connective tissue. I recognize some muscles, the external or extrinsic muscles that attach to the eyeball and would move it around in the eye orbit in the socket. Also on the back, you'll see something that looks much whiter than these yellowy, fatty, and then connective tissues. And that little round white thing right there is the optic nerve coming out the back of the eyeball. Now, there are going to be several layers of tissue in the eye. The outermost, very similar to what we saw in the central nervous system, the toughest. And this would be what we call the fibrous tunic. It's tough fibrous connective tissue. And the white colored connective tissue on the outside we refer to as the sclera, or the white of the eye. The more clear portion, which in a healthy, normal functioning eye would be much clearer than this, it's going to be cloudier due to the preservative and the fact that the animal is no longer living. That's the cornea. And that's continuous with the sclera. It's one continuous layer. Because this is really, really tough, I do not recommend using a scalpel because, again, as we saw with the dura mater on the brain, very hard to cut through that outer layer, and as you're pushing really hard and you finally get through, you're going to put so much pressure on there, you're going to have a little fountain of inner juice. So what I recommend is a scissor with some fine points on it, and I like to try to snip a little opening so that I can get one point of the scissor through there. Maybe poke a little hole. As you can see, this is pretty tough stuff. I have to really cut away. Oh, now I see a little juice, little fluid coming out of there. We'll talk about that fluid in a moment when we get on the inside and take a look. We're going to cut right through the cornea. We're going to go all the way around the eyeball, gently. Remember, it's a little package with delicate things inside that you don't want to harm, even though it's pretty tough. Okay. And then we got to go this way. Oh, that is tough stuff, I'll tell you. And we're going to kind of open that up like you would open an egg. Whoop! Look at that eye that popped right out of there. Okay, so we should see several interesting things here. The very liquid or watery material was found in the cavity here, what we call the, of course I can't get to it. Okay, we have the anterior chamber here and the posterior chamber back here. The dividing point would be this middle layer of the eye that contains the iris, in this case up in the front. And that iris, which is made up of muscular tissue, is continuous with the middle layer of our 3 layers, which is called the choroid. And in that front anterior area is the aqueous humor, or very fluid, watery material. In this larger posterior chamber, we have this gelatin-like material called the vitreous humor. Vitreous means glass, and so it's sort of glassy looking and very gelatinous. And that, you can see this right here is the lens, which looks white from the preservative and lack of circulatory flow. This one would be clear in a normal healthy eye. But that lens, we'll take a look at that a little bit more closely in a little bit. And then you'll notice this pattern on the front that has little lines radiating out from the center is an imprint with pigment from the inside of this eye. And that imprint comes from the shape of the muscles that are part of that iris. So we're going to cut that out and take a look at it. We're going to see 2 types of muscles surrounding that are part of that iris and surround the opening in the middle, which is called the pupil. Okay? And what you hopefully can see, if the light, is the light good there?
Yep.
What we can see is that around the middle part of the pupil are circular muscles that go all the way around the opening. And then attached to the outer edge of that are some what we call radiating or radial muscles that come out. Okay? If the muscles in that circular layer here were to get shorter, they would make the hole smaller. So the circular muscles are constricting, and they constrict the pupil. The radial muscles, when they get shorter, pull those constrictors out of the way, and so it makes the pupil larger. So these are your dilating muscles, the radial ones. Okay? I like to think of them as like rays of the sun. See the pattern of like the rays of the sun on there. And then the other thing you'll notice in here, in this chamber where we have the pigment, something that we don't see very often, we don't see in human eyes, and we only see in some animals, particularly animals that have better nocturnal vision than we do, is a structure called the tapetum lucidum. It looks a little bit like, I'm going to push this creamy colored white material, which is retina, aside, and we'll get to that. We'll look in the other part of the eye at that retina in a moment. But what you can see is this beautiful iridescent turquoise green area. That actually helps animals trap more light inside that chamber to help them see better at night. And that's why when you flash a light in the eyes of a deer or a dog or a raccoon or a cat, it'll reflect back at you in usually different colors for different types of animals, yellow, green, orange. And that's that tapetum lucidum. And that's why human eyes don't do that, I guess, unless you're a vampire or something. And then, so apparently we have some interesting eye things. Now, this creamy stuff that I moved aside was our innermost layer, what's called the neural layer. The choroid layer, which is this middle layer and which has the pigment on it and which is continuous with the muscles, the intrinsic internal muscles that regulate the pupil. That layer is referred to as the vascular layer. And the innermost layer is referred to as the neural layer, is basically the retina. And the retina is very, very delicate. And one of the roles of this vitreous or glass-like humor is that it pushes the retina up flat against the back of the eyeball, kind of like if you were trying to project a movie onto a sheet on the wall and you want that sheet to be nice and flat or your images would be distorted. This is your sheet, and that vitreous humor presses that sheet out, and you can see if I just disturb it a little bit. So if someone were to have a blow to the head, a significant blow to the head, it could disturb this enough that it would wrinkle like this or shrivel up. And that's called a detached retina. So a bad head injury can cause that. Sometimes can cause permanent damage. Sometimes can be repaired. What you'll notice is that it just slips right off of that choroid layer, except for one little spot. I can't seem to get it off this one little spot. That one little spot where it's attached is the spot where all of the axons from all of those nerve cells are going to exit the eyeball into the optic nerve. And so if I pull on that, you can see that little round opening, which on the other side is there's that optic nerve we found earlier when we were looking at the external part of the eyeball. This white structure right there is connected to that. And that's your blind spot, because in that spot you only have axons, no receptors. That means you can't see right there. And so that little
bit.
That little blind spot right there, that little spot where that's attached, has no receptors, just axons. And so therefore that's your blind spot. Your brain learns to compensate for that in your images. So you get enough information from the surrounding area that you don't notice that you have a blind spot. Your brain helps you to not notice that at all. The last thing we're going to take a look at is the lens. We're going to kind of separate this lens off of here. When I was a younger person many years ago, we were working on frogs. They're so hard, so bouncy, those little lenses. We used to play marbles with them. But one interesting thing that you'll notice, this is where you might try and use a scalpel, but you have to be careful because it is round. You know how it is when you're cutting peas. Shoot across your plate. If you cut into it, what you'll find is it's a lot like an onion. Notice I make a lot of food references, in that it has lots of layers inside there. Now, I don't know if we can see that real well. Let's see if I can grab that with a forceps. Maybe it's a little slick. Let's see here. Okay, get the light on that just right. It has layers of protein. It's just special proteins in that lens that are layer upon layer upon layer. And this is what focuses your image. And this lens is also connected to a set of muscles within that choroid layer, which are attached to some special little ligaments called suspensory ligaments. And we call the little batch of muscles at the ends of these suspensory ligaments, suspended right here behind the pupil, we call that the ciliary body. And that ciliary body, when your brain is trying to help you focus, it will pull, and that will make the lens stretch and get a little flatter, and that will change the position of the image that you're looking at. And if it loosens it up, that image moves a little bit closer, so it tries to get it to hit right on the back of that eyeball where the retina is. And if you don't do that very well, then you might need glasses. The glasses would then correct that distance in order to make it sit right on the back of that, right on the retina, your projector screen, so to speak, on the back of your eyeball. The other interesting thing about these proteins in the eye is when we go back and we look at this cornea, so usually the retina would be sort of clear, pretty clear, and the cornea should be clear. They can get cloudy as you get older or from certain injuries or diseases, and you can get what are called cataracts and have those replaced or have the cataracts broken up. The cataracts are crystals that form in here and cloud up these layers. But the material that's in the sclera is exactly the same material that's in the cornea. Why is this one white and this one clear? And that's because of the way that the fibers are aligned. And the fibers in the sclera are very random, and they're kind of a reticulum or a mesh-like network where they're all going different directions. And that makes it opaque. No light can get through. But it would be kind of like if you were taping over something and you put lots of tape over there because you don't want to let any light in. Over here, the fibers all run in one direction. They're all parallel, kind of like Venetian blinds. So then that allows light in between those fibers, which gives it the appearance of being clear. But basically it's the same kind of material. It's a collagen for the most part. And keratin, structural proteins that are sturdy. And there you have a sheep eye.
Now, this is the only part of the human body that doesn't get oxygen from the blood supply. Is that correct, the cornea? Or is the whole sclera?
Yeah, there's there's a few places in the body that don't get good blood supply,
okay.
but where you have dense regular connective tissue, you do not get generally great blood supplies.
Okay.
And that's why those things are very hard to heal, like ligaments, tendons, cartilage has very little blood supply, and then the sclera has no blood supply.
Okay.
And actually the cornea doesn't either. It's interesting when somebody gets a red or a bloodshot eye, something we can't see on this sheep eye right now. When it's in the sheep, there'll be a very thin layer. In other words, the skin that's on the sheep's face, and same thing on a human, comes down over the surface of the cornea. Just a thin layer of skin. It's so thin that it's clear. We call that thin layer of living skin. It's continuous with your eyelid, under your eyelid.
Wow.
We call that layer the conjunctiva. And when that has blood vessels in it. And when somebody gets an infection in that or it gets irritated, then the blood flow increases in there to bring, to heal. And so they'll get a bloodshot eye. They'll get a red eye. So the redness is from the conjunctiva, not from the cornea or the sclera itself.
Okay.
Sound waves are gathered by the pinna and directed down the auditory canal to the eardrum or tympanic membrane. The mechanical energy of the sound causes the eardrum to vibrate. This in turn causes vibration of the three middle ear bones, first the malleus, then the incus, and finally the stapes. These tiny bones amplify the vibrations. The stapes comes in contact with the inner ear at the oval window. The fluid-filled inner ear is a coiled structure called the cochlea. Vibrations conveyed to the fluid cause bending of hair cells. If we examine this cross-section of the cochlea, the tectorial membrane can be seen at the top. In it are embedded the tips of hair cells, which are shown circled in yellow. When the hair cells bend in response to vibration, the rate of firing by the neurons at their bases changes. These signals are conveyed to the brain and perceived as sound.
Sound waves strike the tympanic membrane and cause it to vibrate. Vibration of the tympanic membrane causes the three bones of the middle ear to vibrate. The footplate of the stapes vibrates in the oval window. Vibration of the footplate causes the perilymph in the scala vestibuli to vibrate, which in turn causes displacement of the basilar membrane. Short wavelengths from high-pitched sounds cause displacement of the basilar membrane near the oval window. This movement is detected by hair cells of the spiral organ, which are not visible in the animation. Long wavelengths from low-pitched sounds cause displacement of the basilar membrane far from the oval window. Again, this movement is detected by hair cells of the spiral organ, which are not visible in the animation. When the vibrations reach the perilymph in the scala tympani, they travel to the round window where they are dampened.
In this video, I want to discuss some of the structures of the ear with the outer ear, the middle ear, and the inner ear. So in this model, we can see a major portion of what is called the petrous region of the temporal bone, and then on the outside of the skull, we can see the auricle of the ear, and it is divided into several different regions. We have the helix, and on this model you can see helix, a little bit of the tragus, you can't really see the ear lobule, and you can see the external auditory canal. Notice the canal goes through soft tissues and then through the external auditory meatus of the temporal bone. The external ear starts at the auricle and goes up to this structure here, which is our tympanic membrane. The tympanic membrane is the wall between the outer ear and the middle ear. On this model, I can remove just temporarily the tympanic membrane so you can see the size of the tympanic membrane, commonly known as the eardrum. Put the tympanic membrane back in, we can see there are 3 bones associated with the tympanic membrane: the malleus, the incus, and the stapes. If I rotate this tympanic membrane laterally, I can see attached to the posterior surface of the tympanic membrane the malleus. Articulating with the malleus, we have the incus, and then articulating with the incus, we will have the stapes. On this model, they have the stapes attached to the inner ear at the oval window. So here we can see the stapes. I'll lift the inner ear portion out and show you the stapes attached to the oval window. So here we have the stapes. Putting the model back together, we can see tympanic membrane with the malleus, incus, and in the background the stapes. Tympanic membrane begins the area known as the middle ear, also known as the tympanic cavity. The tympanic cavity has an opening from the nasopharynx known as the pharyngotympanic tube. And here we can see our pharyngotympanic tube traveling up to that middle ear cavity. Remember, from the pharynx to the tympanic cavity, hence the name pharyngotympanic tube. Immediately above that pharyngotympanic tube, we see a muscle traveling along inside the petrous region of the temporal bone. That muscle continues on here on this model as this little dark gray piece. That is the tensor tympani muscle. The tensor tympani muscle functions to dampen sound vibrations in the ear by pulling the malleus away from the tympanic membrane, the tensor tympani muscle. The rest of the structures on this model are structures of the inner ear. Now, the inner ear is embedded within the petrous region of the temporal bone, and we see this better on another video, but I'll lift this portion and we can look at it just briefly. We can see on this inner ear the 3 semicircular canals, the area of the inner ear known as the vestibule, and then the area known as the cochlea. If I turn this portion around, we will see that coming from the cochlea, we have the cochlear portion of cranial nerve 8, the vestibulocochlear nerve, and this here is supposed to be the vestibular portion of cranial nerve 8, the vestibulocochlear nerve. We cannot see the oval window on this model because they left the stapes attached, but we can, if we turn it up, see the round window. So you want to consult the video of the inner ear structure so we see all of this in an enlarged fashion. The last thing I want to point out on this model is this structure right here. This is our internal carotid artery as it passes through the carotid canal of the temporal bone and then passes into the cranial vault. So our internal carotid artery passing through the carotid canal of the temporal bone.